Good morning, everybody. So I'll be talking more on the computational side because the Congress is of the World Congress on Computer Science Applications. And the theme is on doing parallel high-performance computing for extracting signals from the noises. The noises might not be a noise to you or to someone else, but you have to define what is noise for you and what is not noise to others to get the signals that you want. So the talk is Para-Seqs, parallel sequence analysis, in the context of next generation sequencing technology, which is the hot area in the last decade, if not last 3 years, right? And the sequence pattern match that has been deployed is, for instance, taken the gene sequences for the prokaryotes, which are the lower organism. I won't get into the biology of it, even though I know it. And the eukaryotes are usually the broad definition is the higher organisms. They're more compartmentalization in the cells and stuff like that. So I'm currently a faculty at the University of Virginia, UVA. I'm also affiliated to Leipzig University in Germany. But this work was done a bit earlier, so that's why I haven't put the UVA over there at the moment. So the data economy talks about having huge data, taking a funnel, passing it through a pipeline, doing filtering, processing, and in the end, getting information that you want. The approaches to them could be either rule-based. Rule-based means that you are either a domain expert yourself, such as in our current context, the biological sciences, you're already a biological scientist, and you're applying data science techniques on it, or someone who is a biological scientist, such as a domain expert, and get information from it. So rule-based is, or the science of it, the ab initio method for knowledge that already exists, such as we already know the polyadenylation site. Yeah? We don't need to write a data science algorithm for it. We just know that this is the information I need to map into my code and get the information what we want. Right? And there are many such examples for which the knowledge exists, capturing the knowledge into the code to get the data that you want. The second method is the classifier methods. Classifier methods, or the homology method, comprises of using a tool which is custom made for a certain kind of problems. It can only be used for that kind of problems, that kind of data set. And the way it works is it makes a model of it and then goes for matching the models of similarity, or homology that you call it. That's the second way to do a data economy, data extraction mining method. And the third method is the most popular in the last 3-4 years. And it has been really the talk for all the conferences in this Congress, is the data mining. Yeah? The data mining for which there is no existing model. You have certain algorithms, call it deep learning, call it machine learning, call it artificial neural network, call it k-means support vector, call it there are many variants of these things. And you can use them, and they have certain ways to use it. You have to only take your data and put it into the format that it uses and use your creativity and apply it. So this is the whole, broadly what a data scientist, such as a bioinformatics data scientist, can use it. And a good bioinformatics data scientist would, not just bioinformatics, any data scientist would combine all these information to the modeling process. So just since we are in a BioComp conference, I'll just give a central understanding for those who are not the biologists. The concept is you have a DNA that's stored that has more or less a permanent information, and whenever a certain information is needed, the cell extracts a certain section of that into RNA, transcription it's called, step 1. Then RNA is then coded into step 2, which is protein. These are very broad definition, and the proteins are the functional unit. It can be a structural component, or it has a phenotypic component of it. And the fourth pathway, the complementary DNA by reverse transcriptase, is also gaining popularity in the human context. It was already popular in the viral context. There were retroviruses and stuff, but these are also getting really popular these days. Ever the post-genomic sequencing era has become very, very transparent. And what is done is, this thing is, everybody knows, it looks very confusing to someone who sees for the first time, and that's why it's called confusion matrix. It essentially does a sensitivity and a specificity analysis. So if you already have known information, you know what is the result that is true, what is the result which is false. So you can model your way, so this is the actual, this is predicted. So you can model your way so that there will be a combination of where true positives are, true negatives are. So this is what you wanted. And the off-diagonal, the reverse diagonal will be the ones that you don't want. So sensitivity and specificity is a very key characteristics of any data analysis that you do eventually. As an example of classifiers where there is no deep requirement for cooperating or collaborating with a domain scientist, would be a prokaryotic gene finding algorithm. There is no complexity. We know that there is a DNA sequences. There are 6 frames, 3 in the forward direction, 3 in the backward direction, depending upon the codon positioning that you use. And there is a start codon that is known. There is a stop codon that is known. If you just try to find those signals, you can simply get those patterns, right? And if you move to the higher organism, you just can't take the data and push it into the algorithm and say, hey, give me the results. You have to talk to a domain specialist, the molecular biologist, modern biologist, to know what are the complexities involved. It's not just a piece of fragment that comes out. It comes in many ways. And there are already known informations of what's the acceptor site, what's the donor site, how many exon exchanges can happen, which all exon can comprise in the final output, or maybe at sometimes in a rare instance, an intron can also be finally translated. All those possibilities have existed in the literature database, right? So this is an example where just data mining by pure methods, by a pure computer scientist will not work. You have to take into the consideration the collaborations with the domain expert. And that's the focus of this talk, where I've been talking mostly about the gene sequences. And of course, the F-scores and all those things will definitely hold good over here. Going more into the science knowledge that is there, the more information you can get, the more better it is for the data scientist to model it into your algorithm. For example, an influenza virus genome will comprise of 1 gene per 1,000 base. Whereas if you go to higher organisms, such as human beings, well, there may be more higher organisms, but let's say human being is one of the top highest organisms. There's 1 gene per 100,000 bases of the genome. You see, the density varies. This information, a pure data scientist will not know. He will have to, do some literature mining, consult with domain experts, and incorporate into the coding that is done. More such examples are that of the gene, the total number of genes that we know in the prokaryotic databases compared to their length, right? We can clearly see that more are converging towards the shorter side of it, at the maximum at 8,000 bases. Remember this number, 8,000 in prokaryotes. And there is not a uniform distribution of the genes to their length. When you go compare the same numbers to the eukaryotes, there's a vast difference. You can't use the same algorithms. There's a vast difference. The total number of genes versus the total length of the coding region is 50,000. Yeah, that the previous one we had 8,000, here it's 50,000. And the distribution is more or less uniform here. So you see, the pattern has varied, and this information is new information that has to be incorporated into the workflow. A bit about the high performance computing, the grid computing, you call it grid cluster computing and stuff like that. Or these days you also have GPUs and maybe ARM cluster and these kind of things. So there's a whole variant of this domain is like evolving on its own. To a very beginner, you can think of it as a data set. You partition it into segments and push it into different computing cores or CPUs in layman terms, and then you get the results. But I mean, this is the thing can actually get very very complicated. That was a bag of task approach, where you have a bag and you throw the jobs to the processors which are free. And this can be a more complex way, that when you start with all the processor computing, and then you realize, oh, this particular information that has been processed needs to match with the next processor. And there are a lot of task dependency there. An example is the fast Fourier transform that I've given. Of course, I've not used here, but I've shown this as an example. So coming to the ORFs, which are the loose term for the gene open reading frame. It has the upstream components like promoter, downstream components, and many of the things that are there. The way I did it is that you have you load the FASTA format file. FASTA is a format in the sequencing. There are FASTQ formats as well. And then you can already, if you have the knowledge of the coordinates of known genes, then you can keep that with you, and the data can be split based on the information that we already talked about, right? The data can be split. The region where we split, that has to be split in depending on which organism we are talking about, right? So the data initial gene we store it there. Then, if the data is already split, that information can be taken by each of the different processors and matched to the different split data, right? And so this all goes in parallel. We extract sequences and store it over there, and intentionally put a barrier synchronization. What a barrier synchronization does is that if a computing core finishes faster and another one is still running, then it will ensure that all of them waits until that section of the programming has completed. This is a very like a safety measure that is usually taken, but it should be, it's a suggestion that this should be removed later wherever possible. It is, for the beginning part, it should be done like this, because there are always overhead that is associated with it. Then once the coordinates of the splitted files have been extracted, you can concatenate them, combine them, and once the combined long open reading frames have been obtained, you can then run your artificial intelligence, machine learning, whatever tool you want, you can run there. I've run interpolated Markov model over here, and that's what has been the tradition in the gene search algorithms. So that has been used here. And then to run, once the model has been created, for that you need to do it only on one core, because it's not a splittable, not an easily splittable task, I would say. You can split it, but then the accuracy can reduce. You don't want to reduce the accuracy. And then from there, you can, once the model has been generated, you can push this model into the rest of the genome where the gene sequences or ORFs are not known, right? So get the corresponding gene sequences, extract in parallel, and in the whole process, there will be a lot of temporary files that are generated, which you don't need, you can remove it at the end. So this is like the algorithm, but when you actually implement it, it will be a really painful job, especially when you want to increase the speed up. It looks very simple, implementation takes a lot of challenges. So here's a mind-blowing performance that you see. Why did we do all this? Why? Why do I need to think about parallel processing and these things? The simple answer is time. Time is our enemy. If you run the whole code, this whole thing, I can run it on giving one core. It will take 1.5 years or more for just one chromosome of a plant test, Arabidopsis thaliana model plant. One chromosome, it will take 1.5 years. Of course, I didn't wait for 1.5 years. This is estimated. I had to kill the program because it was telling, after 3 days, it was telling me it's going to take 1.5 years. Right? 90,000 minutes. And by increasing the cores to 4, just 4 times more, there's a significant reduction in time in terms of minutes. And actually, I ran it till 190 cores. And you can clearly see that after 10, you can't even see by the human eye if there is any reduction. Yeah? And to make it visible, I have to make a log plot of it. Not that it is any mathematical advantage to do a log of it, but to make it visible, I have to do a log of it, and so that you can see the logarithmic decay in the time that with every subsequent addition of a core. And usually with any high-performance computing system, whether private cloud or public cloud, or even if you use GPU, which comes with several cores, you have those things available. The challenge, of course, is a bit of data transfer, but those things are also point-to-point transfer things people are coming up with. So to show more of the results that this tool was worked upon, I use it for Plasmodium falciparum as well. And here you will see there is not so much significant drop in time, execution time. And this is the result I'm showing you, because it's not like a black magic, it will happen on its own. It also depends on the data complexity, right? So the data complexity of Plasmodium falciparum is far higher than the previous one that I showed. Robustness. Robustness is a major criteria that if I am doing something, and if I set up a business or if I set up collaboration, I will get a lot of data that will come in, and I should essentially be able to partner up with anywhere in the world. And as long as I have the data, I should be able to do the things with the same pace, right? So if I increase the data size for the same quality of data, by 2 units, 3 units, 4 units, the time taken should not go exponentially. It should go linearly. And that's what the robustness characteristics of this slide is capturing. Then the slide is not visible, but this has been done for different kinds of data set or organisms. So that was for eukaryotes, and that's where the real application of these kind of tools are in the next generation era, where the next generation sequencing can be first compiled into a genome, right? You have sequences which are 70 bases or maybe 200 bases, 300 bases, but they're not definitely 3 billion bases, right, which is the human genome, right? So if you have the whole genome, if you make it as an assembly, you can then pipeline into techniques like this. And forget about the next generation tools and stuff like that, which are with lots of problems. You just parse it into a parallel processing framework like this. And that was the application where I showed the eukaryotic genome, which are the bigger genomes, but it is definitely wonderful for prokaryotics as well, as I showed it for different organisms too. So those were so if you're coming from the parallel computing world, there is a distributed computing where they don't share a common memory. There's a shared memory parallel computing. So those are called symmetric multiprocessor, where usually OpenMP or POSIX threads are used. So I implemented the whole algorithm on the SMP version as well. So these are the performance of the SMP version, as you can see. And I also wrote a book with some bioinformatics techniques and stuff like that as a chapter in a whole genetic engineering book. So in case you're interested, you can also just have a chapter of it. You can just mail me, I'll send you that chapter, if not the whole book. Okay, that ends precisely my talk. That's my webpage, personal webpage, and it's my email address. And please ask me questions. I would love to answer them. Yes, please.
It's very important work.
Thank you.
In my world, we have a somewhat different problem, and that is the world of clinical medicine, specifically clinical cardiology.
Sure.
To give you an idea of the magnitude of the problem, if one were to monitor 5 different physiological parameters in a patient for 3 days, which the values might change with every heartbeat, which they often do, in 3 days from one patient, one would accumulate 1,740,000 data points.
Sure.
We have monitors all over the place in hospitals, and yet most of that data is ignored. Not because people are lazy, but because they can't possibly assimilate all of that data. And it's not necessary just to look at one parameter. It's necessary to view changing relationships among different parameters. And so a big part of my effort, and I think could be an effort of people who are far more sophisticated in computing than I am, would be to provide methods of display that enable us to waste far less of that data. Just, I'll say one more thing.
I already have 3 questions in my mind for which I have the answers for you.
Okay. If I came to this conference next year, and even though I probably would not remember your name or the institute with which you were affiliated, I would probably realize that I saw this face before.
Sure, thank you.
The ability of the human eye and the human brain to recognize patterns is stupendous. And so one of the tasks of people like yourself could be to put data in recognizable patterns. The eye and the brain will do much of the rest. That's the point
Sure.
I was...
Yeah, I mean, it's a very good point. I will try to break your long question into short pieces. I mean, there are lots of techniques in data. There's more experts in data science than I am. There are lots of techniques like time series. If you have a continuous flow of data, time series is a wonderful way, ARIMA modeling, and a lot of things are there by which the data can be modeled and presented. And presentation, visualization, I totally agree with you, is a key component. And it's not a very difficult task. You just need to plug in things, and only the sections of the data can come on the visual portal. And there are techniques like PCA for dimensionality reduction, but I wouldn't push too much on the PCA, which is principal component analysis, too much, given that I am pushing more the high performance computing, because all I care is that give me all data, I'll process it. I don't want to reduce the dimension and lose 10–20% of the information. But
Okay.
yeah, the point is taken.
Thank you very much.
I think that gentleman has one more
We need
question.
to close it because it's